Hello, thank you for joining us for our chromatography webinar, Peptide Purification Overview. If you have any questions during the webinar, please utilize the chat or Q&A functions within Zoom. All questions will be answered at the end. This webinar will be recorded and a video will be made available in the next few days. Our presenter today is Jack Silva, Application Chemist. With that, I will turn it over to Jack.
So why would you use a mass spectrometer? It saves time and solvent, so you know when the compound elutes. You can terminate on target so that it stops the pump, so after your peak comes out, you can save time and solvent and just jump into the next run. And as I will show you, it will allow positive verification of the purified peptides. The conventions here, M is the mass of a given molecule. The base peak is the tallest peak in a mass spectrum. The M plus H positive is the mass of a molecule with a proton, and it carries a positive charge. And it's possible to have multiple charges so that you have M plus 2H or 3H, and you can have multiply charged species, and those are very useful. You can also see M plus sodium or M plus potassium, or adducts such as M plus solvent plus H, where the solvent is the carrier solvent from your mass spectrometer, or your HPLC solvent such as methanol or acetonitrile. Okay, some definitions. Nominal mass is the integer mass that we use in conversation. So our friend over here, which is caffeine, has a formula weight of 194, and that's the nominal mass. Its monoisotopic mass is similar to the exact mass. The exact mass is used by some chemistry software. It is the sum of the atomic mass of the most common isotope. And the average mass is the sum of the average atomic masses in a molecule. So why are we concerned about this? For small molecules, you generally want to use the monoisotopic mass for detection because that matches the peak. Here's our caffeine, and then we have a couple of isotopic peaks out over here. Its average mass is actually a little bit larger because of these isotopes. Now let's look at a fairly small peptide, this ACE inhibitor, whose monoisotopic mass is 1091. Its average mass is 192.2, or an entire Dalton different. And that's enough difference to miss the mass detection in extracted ion detection, which we'll talk about. Next slide. The mass spectrometer will see the average mass of a peptide because those molecules, even the small one, is large enough that it will have a typical average isotopic composition to it. And I usually start using the average mass around 800 grams per mole or a bit larger. And very large peptides will show a much greater difference between the monoisotopic and the average mass. I just mentioned extracted ion current, and what that is the ion current for a limited mass range, an M over Z range of 1 to 2 Daltons, although that could be just a little bit larger. And it's used to monitor characterized compounds when you're running your chromatogram. The total ion current uses very nearly the entire spectral range, and it's useful for natural products or if you just don't know what the compound is. And I sometimes use it if my molecule generates a very weak molecular ion peak. I'll run a range that encompasses the fragments, or I will show you how I use a small range to look at all of the isotopes in a peptide. So first thing you would want to do is verify that your peptide is there and determine the preferred detection ion. So our example here is thymosin, which has a mass of 3,066 grams per mole. My mass spectrometer only goes up to 2,000 Daltons, but I can detect the peptide with multiply charged ions. And I can see the M plus 2H, M plus 3H, and M plus 4H. And the presence of these multiply charged ions simultaneously in a peak is unambiguous proof of the compound that you want to purify. And you can look for these using MS method development on our systems over here. These multiply charged peptides are charged via basic residues, no, your amine groups. There's some other residues that can pick up a proton as well. Occasionally, the amide linkage does as well. And this equation over here allows you to calculate what peaks should appear in a peptide as multiply charged. So within a single peak or a single method development, if you see peaks that fit this equation, where M is the mass of your molecule, and then ZH is the number of protons, and you divide by that same number, Z. If you see that, that's unambiguous evidence that you have the correct compound. Now, how many charges can we put on? It really depends on the length of the peptide, the number of residues that can take a charge. Not every residue can. And also, this paper down over here suggests that you need 3 residues between a charge. So you can't have, say, 2 basic groups right next to one another and put a proton on both of those. Another example is this AS48, which you can copy that down. It's 7,787 grams per mole. And it shows up over here on our mass spectrometer. Again, 2,000 Dalton limit, but we can see the M plus 5, M plus 7, M plus 8, and even an M plus 9H over here. So the multiply charged species allowed to mass the charge to fold into the range of the pure ion. Okay, so now I'll use the mass spectrometer to help describe method development. And it's very easy to go from analytical to prep. There's a paper that we have from ACS Combinatorial Science that uses the time on target algorithm. What you can do is on your prep system, use a model compound and set an analytical, set a prep run and determine that your compound elutes at 50% solvent in this example. I then use an analytical column with matching chemistry with the same solvent system that I used over here. I can then run a scouting gradient on my analytical system, and wherever the peak comes out, I assign to that same solvent composition that I used up over here. The difference between the 2 gradients is an apparent gradient delay. So, running a peptide on an analytical system, the programmed gradient suggests that this peptide elutes at 50% B solvent. However, using the applied gradient delay, I see the compound actually elutes at around 21% methanol in this case. Okay, this is thymosin, and the solvent system was acetonitrile water. I see that I made a mistake and wrote methanol there, but it's water acetonitrile, both 0.1% TFA. And then I calculate a gradient on our system, and you see pretty close to 20% B, and it comes out in the middle over here. An easier way of doing this is to use the focus gradient generator built into the AccuPrep. All the calibrations are done ahead of time, and we have an automatic scale-up feature. We have this sample being run, 1635 Daltons, and I used a range to look at the isotope. The water is 50 molar ammonium formate using methanol, And you see that I do a scouting run, and then it calculated the gradient, and it came out very nicely resolved from impurities. Okay, now, how did I calculate this? I started off using acetonitrile water, and I did a scouting run, and my compound came out late over here. However, I was able to raise the scouting gradient up by about 5% and get the compound to elute, focus gradient over here. However, I see from my mass spectrometer that my compound is eluting at the solvent front in the DMSO. I've got a small peak here, and then I've got a couple of peaks out over here. So I have a little issue over here is I see my compound, and but I'm going to lose some of the compound in some of these other peaks that I might not otherwise collect. So why is this happening? Well, TFA is not a buffer. TFA is about 5.9 millimolar concentration, call it 6 millimolar concentration. My peptide loading is around 1 millimolar concentration, and that's enough that I'm not seeing good pH control of my peptide. Okay, peptides are a little different from small molecules in that they seem to be somewhat more sensitive to pH. So I'm going to try buffering the pH. I'm going to use 50 millimolar ammonium acetate at pH 4.5, and I can control that as this is a real buffer. Because I'm using ammonium acetate, I need to switch over to methanol to keep the ammonium acetate in solution. I run my scouting gradient because I changed the solvent and I changed the buffer. And now the focus gradient works first time. I also see a greatly improved peak shape in which everything is now concentrated in only 2 major peaks over here. However, I calculate that this particular peptide has a PI of about 5.5. I'm at pH 4.5. So let's see what happens if I run a lower pH. pH 3.5 using ammonium formate. I further improved the peak shape. I need to rerun the scouting run one more time because I did change the buffer. Now I have greatly improved peak shape. Everything's in one peak, and I'm resolved from the impurities very nicely. Okay, and this is the run that I showed you earlier. I have improved yield because everything's coming out as a single peak, which I can then collect. So the method development summary is method development works best if the peptide is buffered. The buffer sets the pH, and good pH and buffer control can greatly improve recovery because everything is in a single peak. However, if the peptide needs a very high concentration of organic solvent, pH control becomes difficult for a couple of reasons. Is you have less buffer in the solvent system to begin with, and compounds simply aren't ionized in the same way. So your peptide tends to act more and more as a unionized compound. In this case, the mass spectrometer allowed me to see how the compound was eluting. One advantage of flash prior to prep HPLC, we can use flash chromatography as well to purify peptides. Flash is a lower resolution purification technique that uses less expensive columns and equipment. Different selectivities, in other words, the flash column might be different from your prep HPLC column, and that allows different selectivity, and it allows potentially a different order of elution of your peptide with respect to impurities, which could improve the resolution of your compound on your prep system or improve the impurity. So so by removing these impurities, you increase the concentration of the desired compound. Your prep column injection is limited by solubility and sample loop size. By solubility, in your sample loop, you often wind up using DMSO, and that affects your chromatography, and we will discuss that later. We do have ReadySep columns, and they're available in both analytical, flash, and prep. They're all the same chemistry for easy method development, and we have different pore sizes, and we'll explain why that is useful later as well. So flash purification, we have our peptide purified. The sample was dissolved in DMSO because we have this huge amount of nonpolar impurity, and the DMSO dissolved everything. The gradient is similar to the prep HPLC gradient that I will show you shortly. We loaded 100 milligrams, and we were able to recover 40 milligrams from this area defined by the red box. After flash purification, we were able to run it on the prep LC system, and as you can see, it came out very pure. All I did was just evaporated the organic solvent from my flash fractions, and so I didn't need to use DMSO. I got the impurities are very low, and that was almost ready to use for whatever other purposes that you need. Down over here, we see the prep HPLC run. Again, I had to load it in DMSO because of the impurities. That limited my load, my baseline resolution, to 13 milligrams loaded, and I only got 8 milligrams back. The sample from my flash, I loaded 30 milligrams of that 40 milligram sample, and so I got 23 milligrams back, so it's pretty decent recovery. So I was able to greatly improve my throughput by using flash purification to pre-clean the sample. For really, really large samples, we have the TorrentAQ, which is optimized for reverse phase. It's great for multi-gram scale fractions, and we have an optional sample load pump for large and dilute samples. We're going to talk about pore size and chain length on peptide purification. So if we run a small molecule over here and we increase the pore size, that tends to reduce the surface area of your column of the particles. And so what happens is that your small molecules show reduced retention as we go from small to large pore sizes. Okay, and that's simply because there's less surface area for them to interact with. So you have a reduced interaction with the stationary phase, and this is a set of runs with methyl, ethyl, and propylparaben. How about peptides? We're going to use the peptides that we've been using throughout this webinar. And we're going to run them from small to large peptides, 120, 200, and 300 angstrom. And EGFR tended to act like a small molecule. Thymosin tended to act initially a little bit differently. The interaction was increased, and then the interaction got decreased as I increased the pore size. And AS48 also acted the same way. I had increased retention, and then when I went to 300 angstroms, I saw reduced retention of the peptide. So it seems that for these peptides up to at least 8,000 Daltons, 200 angstrom pore is the sweet spot. So why is this? large molecules don't actually fit into the small pores. So the stationary phase inside the pore is not accessible to the peptide. So what I term the effective surface area is now reduced. Large peptides fit into these larger pores, and that increases the effective surface area and giving you better retention. And as I showed you on the previous slide, 200 angstroms is more or less a sweet spot for most peptides that people will run. And the changeover from a small molecule to a large molecule is about 2,000 Daltons in this context. So if your peptide's below about 2,000 Daltons, 100 angstrom pore size will work for you very, very well. If it's above about 2,000 Daltons, you might want to consider a 200 angstrom pore size column. So what about chain length? So let's go back to our small molecules again. As I make the chain length smaller from C18 to C4, again, we see reduced retention. Okay, they interact less with the stationary phase. Going into peptides, though, we find a much smaller effect, and for our EGFR, what we find is very little change for the EGFR. Thymosin, we see that it gets a little bit larger, and the AS48, it gets a little bit larger as we shorten the chain length. So that's kind of opposite of what we see for small molecules. So what we find is that the pore size increases as we decrease the chain length. So we, again, open up the effective surface area of these molecules. There's also a change in adsorption mechanism as you go between small and large peptides. Small molecules can fit in between these alkyl chains very nicely, whereas peptides tend to lie on top of it. And so peptides tend to have a little bit better retention with the smaller alkyl chain. The small alkyl chains also allow for a little bit of interaction with the underlying silica too, where there's some unreacted silanol groups, even with end-capped. One thing to keep in mind is the pore size of the particles are measured before modification. When I say, pore size of the C18 is measured before the C4 or C8 or whatever is added to it. So your pore size is actually a little bit smaller than what we describe with C18. Moving down, sample loading consideration. I see a question over here, and I'll pick up these questions at the end of the webinar. Some sample loading consideration. Weak and strong dissolution solvents. I'm running a flash column over here simply because it shows very clearly what happens inside your prep column. So over here, I took a sample and I loaded it in a rather strong solvent. It's actually 50-50 methanol water. And what we see is as the compound starts to, as the column starts to run, the compound runs down the column, and you see this little peak over here is actually some of the compound eluting very early that got dragged down by the strong solvent. You also see that the peak shows a little bit of fronting over here. Again, you can see that in the picture over here. And if I dissolve the same sample in water over here, at the same time I took the picture in this first column, you see that the second column doesn't show any compound, and a little bit later in the run you see that there's a very nice band over here, and that's reflected in the chromatogram where I have a very nice symmetrical peak over here. Okay, so let's move on to DMSO, which is very common to dissolve many compounds. I have a polar compound over here, and I dissolved it in DMSO. And all runs here will use the same concentration of compound. So the left was a 1 mil injection, and I have nice baseline resolution. The peaks aren't exactly beautiful, but they will serve very well for prep chromatography. But because I have that baseline resolution, I said, well, let me try injecting more. So I injected 2.5 mils, and now I get very bad chromatography over here. The peaks run into each other, and they run up into the DMSO. While DMSO is very easy to dissolve most compounds, DMSO is still stronger than water, and in fact, it's stronger than 20% water, which is where I started my gradient. So it does act a little bit as a chromatography solvent, and is a somewhat of a strong solvent, as we saw in the previous slide. So here's the 2.5 mils of DMSO. Now I dissolve the sample in 2.5 mils of water at the same concentration, and now I have baseline resolution again. The peaks have a typical shark fin indicating that the column is overloaded. However, as long as I have baseline resolution, that's fine. I'm not doing analytical chemistry, I'm doing prep. I then ran the sample, same concentration as above, except I did a 4 mil injection, and I still barely have baseline resolution, and it greatly improved my throughput. I improved it by almost 4-fold over my earlier run by switching over to water. Okay, so that's how come flash purification helps, is it lets me remove impurities that prevent me from dissolving my peptide in a much weaker solvent. Okay, some dissolution strategies over here. My calculated gradient shows the compound elutes in 20% or less B. Try dissolving a peptide in water. If your calculated gradient suggests that your compound elutes at greater than 50% B, DMSO is usually okay. If you're somewhere between 20 and 50% B, you could take your DMSO and you could dilute it with a bit of water, and you can maybe dilute it with quite a bit of water and use the simple load pump to load the sample. As long as it's in solution and is in a weak solvent, generally the sample gets concentrated in a nice band at the head of the column. And this sample load pump is also pretty good for diluting, for desalting samples as well. So with that, I do have a question over here, back to pH. Since the remaining silanol groups act as an acidic moiety at medium pH 5.5 and become ionized, do I think that the remaining free silanols are important, or is it linked to the mobile phase pH itself? That's a good question, and there might be some effects of the linked silanol groups, especially if you run, say, a short chain such as a C4 or C18. So I would not necessarily rule that out. Another question that I see over here is, since flash purification increases the analyte load of peptide over the sample impurities, can I reach a point where the impurities cannot be detected? I would say that it depends on the peptide. The flash purification that I did earlier worked very well at reducing the impurities greatly, and for many purposes, that peptide was probably ready to use just off of the flash column. So any other questions, please? Go ahead and type them in. And again, I thank everyone for your patience earlier, and I'm sorry that we had such trouble getting everything started between the screen freezing up and the sound disappearing. How does chromatographic behavior change as we get closer and further from a peptide's PI? A peptide's PI, what it means is that you have all the charges on a peptide. Well, let me rephrase this. The PI is defined as the pH where the net charge of a peptide is 0. Okay. What that means is that it doesn't mean it's a neutral molecule, so to speak, but just the net charge is 0. And you could still have multiple charges. So I like to go away from the PI one way or the other just to force the molecule to be either all positive charged as possible or all negative charged. At the PI, you've got charges moving around, and I think that you'll have multiply charged species. There's someone who is asking why can more be loaded onto, I guess, a flash column. The reason why I can load a lot more on a flash column is the column is sometimes rather large, and I'm not trying to do an absolute purification. I'm just trying to clean out some late eluting impurities, really. So that means that I'm just trying to make it easier to run on the prep system by removing some of the impurities and also by cutting out some of that of the late eluting stuff. I was able to greatly improve the purity of the desired compound. Does the flash purification affect the activity of the protein as a enzyme? Well, it should not. It's not going to affect it because it's the, in our case, it's the same C18 that I used. And again, our peptides are small enough that we don't have a lot of secondary or tertiary structure like a full protein. I have a question about using an isocratic run in purifying peptides. I like to watch the run and hit isocratic when I see my peak starts to run. The reason why I like to use a focus gradient instead of an isocratic is it is very nearly isocratic. It's very close to it. And as for hitting isocratic when your peak starts to come, remember that by the time you hit, you see your peak, it's already come out of the column, and any impurities are coming out right behind it are almost ready to come off the column as well. So you might not be doing very much to improve the purity, but you do increase your runtime. But if it works for you, that's great. But I find very little difference in resolution between a focused gradient and a full isocratic. Another question is, what about solid deposit? I imagine that the question is really about solid load cartridge. I have tried that with peptides, and what I find is this is one exception for flash chromatography where solid load cartridges don't really help the chromatography very much. The reason for that is that the peptides take a little bit of time to come off the solid load cartridge, and I don't get good results using that. How critical is it to match the pH of a buffered sample to the pH of a starting mobile phase? Do they within 1 pH unit or within 2 to 3 units okay? That's a good question. I haven't tried to match the pH when I load my samples, to be honest with you. And I've been getting fairly decent results. If your mobile phase is buffered well and not using like just TFA or formic or acetic acid, hopefully your sample will adjust to the mobile phase pH very, very quickly. Someone's asking about cyclic peptides, and I used to purify those a long time ago, and they're going to behave the same as these other peptides, except that you don't have the both the acidic and basic ends, and so in some sense they're going to act a little bit more like small molecules. Okay, I have another question. Is there an optimal range for focus gradient, such as 5% or less? I like to use 5% because it's very, very nearly isocratic, but allows me some error, like some of the peptides, they are affected by pH, so they might come out a little earlier or a little bit later. So I like to use 5%. A lot of people use 20%, but the focus gradient generator and that time on target algorithm has been generally giving very good results at 5%. And that was an awful lot of questions, and I'm grateful for that.
Yes, well, I would like to thank you all for attending our Peptides Purification Overview webinar today, and thank you to Jack for presenting on this topic. As mentioned, we greatly appreciate all of your patience through our technical difficulties. As mentioned earlier, this has been recorded and a video will be made available. If you have any questions regarding this webinar, please feel free to reach out to me through Zoom and I'll forward along to the appropriate person. Thanks again for joining us. Stay healthy and have a great rest of your day. Thank you.
Yeah. Okay, I have one more question. What am I doing if the peptide doesn't dissolve in anything or only in TFA? Well, it's an odd peptide that doesn't dissolve in DMSO. If it only dissolves in TFA, I suggest that you're going to have to inject it in TFA, and you're going to have to, I suggest that you run an analytical run to make sure that the peptide does come off the column, or use a small amount, and you're going to have to do method development in a more traditional way. If it has no charge, I suspect it's going to elute in like very high acetonitrile also. So you might be running it without buffer, with very little buffer, or at most just some TFA. Okay, I thank you all for your time, and I thank you for your patience again.